Okay, so this lecture will include a quick tour of the brain. We'll speak briefly about what brains do. We'll discuss some methodologies that are used to manipulating and measuring activity in brain areas. And we'll speak about neuroplasticity, what it means, and how can we utilize that in neuroscience research. What is neuroscience in general? Any ideas? Just forget about the slide. Whenever you heard the name of neuroscience, what does that tell you?
Study of the brain.
It is the study of the brain. Nervous system. What else? Other ideas, like brainstorm. I mean, whenever you think about neuroscience, what particular figure, what particular picture comes to your mind? What particular term?
Biology. Neuron.
Neurons, cells, medicine. Okay. So we have different perspectives of what neuroscience is. However, people in the field define it as the scientific study of the brain and the rest of the nervous system. So it's not only the brain. So neuroscience is not only the brain. There are many other things that are considered neuroscience that don't lie within the boundaries of the skull. Okay. So saying that, or that being said, let's start with the brain. We know that it's not only about the brain, however, we will start with the brain. So what are the major divisions of the brain that we're going to be discussing today? Okay, as you can see here, in general, the nervous system of vertebrates, and I mean by vertebrates, not humans, not necessarily humans. Humans, monkeys, dogs, rats, are all vertebrates. They possess a vertebral column, okay? The nervous system in vertebrates is composed of two major components. The first one is called central nervous system, and the second part is called the peripheral nervous system. So what is the central nervous system? The central nervous system is composed of the brain and the spinal cord, whereas the peripheral nervous system is anything else outside the spinal cord and the brain. Okay? This particular definition does not really stand for other species other than vertebrates. Because, for example, for an octopus, the brain is distributed throughout its body. Parts of its brain is in its limbs. It's not necessarily localized in a particular area. So this definition only stands for vertebrates. Okay, this figure displays not only the central nervous system and the peripheral nervous system, but many other things, such as muscles and organs. So, I mean, how does that resonate? How does that tell you? What does that tell you about the function of the nervous system?
It controls muscles and body organs and sends signals to them in order to do something else.
Exactly. So this tells us about one of the functions of the brain, which is manipulating these end organs, such as muscles for movement, and end organs, for example, respiration or cardiac function, muscle, the heart, or sweating, or you name it. So, as Yasser suggested, there is one function that we can speak about of the nervous system, which is modifying behavior. Whether you want to term this behavior conscious, such as movement, or this particular function is subconscious. You can't really control it. You can't really, or I would term it involuntary. Okay? So we can summarize the functions of the nervous system as follows. So the nervous system receives information, processes information, and then produces behavior. Okay? So collects information, processes information, and generates behavior. The part of the nervous system that is usually responsible for collecting information is the peripheral nervous system. The part that we really focus on in terms of processing information is the central nervous system. Only the brain? you will realize that it's not only about the brain during this lecture. Okay, so let's take a closer look at the brain. This is a magnetic resonance imaging slide showing us the major structures in a human brain. Okay, so what can you see here is, only roughly speaking, what are the major structures in a human brain. So this part here is called the cortex, and you can see it extends to the front, to the back, to the sides. It's a very thin layer that is only the cover of this of the brain as an organ, and constitutes the major processing part in the brain. What differentiates humans from other vertebrates is that you can see all these folds and all these grooves, and that it's really folded. It's not plain. It's not smooth. Do you know why?
Surface area?
What happens to surface area?
I mean, well, increases surface area.
It increases surface area. Why? Because you need more cells, you need more of the cortex to provide for various functions that are performed by the cortex. Okay? So simply, if you compare this to a giraffe, or a brain of a giraffe, or a brain of a rat, you would realize that the folds, which we call the sulci, are much less, and therefore the surface area is way smaller in other vertebrates, which would correspond to differences in functions of this part of the brain. The other system, or the other part of the brain that is displayed here, is called the cerebellum. In other words, the small brain. And the small brain lies at the base of the brain, and it's really important for coordination of movement and sometimes learning. Okay? The other part that is displayed here is called the brainstem. It is at the central part of the brain, and it serves many vital functions, bodily vital functions, such as regulating cardiac functions, regulating breathing. It also serves as a passageway for all inputs and outputs coming and leaving the brain, coming to the brain and leaving the brain. Okay, and we have also the spinal cord here. That is the second part of the central nervous system. All right. It's not only about the cortex and the rest of the structures that I displayed earlier in the earlier figure, but it's also about many other embedded systems inside the brain. For example, you can see here what we call the thalamus. The thalamus is considered to be the relay station of the brain. Why would I call it the relay station? We would realize later in the lecture that the thalamus receives almost all sensory input coming to the brain and distributes that to the cortex. Therefore, it's a very vital structure in the brain. Other than the thalamus, there are many other structures, such as the basal ganglia, which is a little bit next to the thalamus in this area. We also have the hippocampus, which lies roughly here in this area, covered by the brainstem here, and the amygdala at the tip, at the anterior tip of the hippocampus, which is also important. The hippocampus is important for memory formation and storage, while the amygdala is very important for emotional processing and emotional memory. Last but not least is a number of cells, of neurons, of fibers that we call, that actually forms a passageway between the two hemispheres of the brain. So in almost all vertebrates, there are two hemispheres that are very similar in structure and are connected to each other. So what connects these hemispheres in humans is called the corpus callosum. This is a major pathway or fiber pathway that connects both hemispheres and supports the exchange of information between these two hemispheres. Okay. So when I mentioned the cortex, it was only a general rough term, speaking about a very extensive area that is only, as I said, the superficial part or the superficial layer of the brain. However, in order to make it easier on us, once we discuss these particular terms about the brain anatomy and brain regions, it's much easier to subcategorize these areas in the cortex, or subcategorize these areas generally in the brain. So what neuroscientists did is that according to the overlying bone in the skull, they named the part of the brain that is directly beneath. This resulted in a number of what we call brain lobes. We have the frontal lobe, just beneath the frontal bone of the skull. We have the parietal, we have the temporal, and the occipital. Is it really difficult for you to memorize these? Frontal is on the front, parietal is on the peak, temporal is beneath the temples, and occipital is just out there, okay? At the end. Okay. And each one of these particular lobes is associated with a particular function. So frontal lobes are associated with, as you can see here, planning and high order decisions and like very complex cognitive functions and processes. The parietal lobes are associated with touch and feeling. The temporal lobes are associated with auditory processing, hearing. And occipital lobes are associated with the visual system, vision. So let's go back and refer to the quiz. So if you fall and injure the back of your head, which of the following will you most likely experience? According to the knowledge that we just displayed here. Why B? Because you're probably damaging your occipital lobe, and ultimately you are affecting your ability to see. So it is a visual system that gets affected here. As I said, I mentioned earlier, what you see in a human brain can be replicated, or very similar presentation of anatomy is also seen in other species, other vertebrates. So this is what we call comparative anatomy, where you show the different brains of different species and try to compare and match the parts that look together or that look alike or act similarly in other species. So if you look here at the fish, the lizard, a parrot, an elephant. So this gives us some insight into brain function, because if you look at this particular design, there are many corresponding designs within the brain that would reflect in lower order species what are the functions of these particular areas of the brain. And we will speak later about it, but these comparative anatomy lessons taught us a lot in neuroscience. And we could not have accomplished what we have right now without having really lower order animals to look at and understand the basic function of particular brain areas as compared to humans. However, is bigger always better? I mean, if you look at the elephant, its brain is like 5 to 6 folds larger than that of the human. Does this mean that the elephant's brain is capable of more function than the humans? The answer is no, because to my knowledge, elephants can learn, can they or can't they? They definitely cannot learn, they cannot speak. So what I'm trying to say here is the size of the brain does not always count. It's all about the complexity and about the relative size of regions. As I mentioned earlier, it's all about the cerebral cortex that decides what particular higher cognitive functions that we're capable of doing as humans, because we have this capability, because we have that particular anatomy, which is the cortex. Okay. Okay. Another example is that birds in general have larger cerebellum than humans. If you think about the functional specialization of birds, what do? They fly, right? So in order to be able to fly and balance and coordinate movements, it is very important for birds to survive, to have larger proportions in the cerebellar area rather than having higher cognitive function, because this is important for their well-being, right? So what you would imagine is that birds would have larger areas in the brain that serve the function of keeping them alive, or enabling them to do the basic function that they serve, which is usually flying. Okay? All right. So humans generally have a more disproportionately large cerebral cortex, and other animals, other vertebrates that also have relatively large cerebral cortex are considered smart, or considered like higher order animals. Not as humans, but definitely have or possess some functions that are really differentiating them from other species, such as dolphins, chimpanzees. These are considered high order animals because they have, relatively speaking, larger cortices that enables them to possess higher cognitive power. Okay, we spoke about the brain so far, right? General description, general anatomy, major landmarks. What is the brain composed of? What is the building block? Okay, white matter and gray matter. So what is gray matter? What is white matter?
The gray matter is the myelin.
Or, so the white matter, it's fine, no problem. So the white matter is the fibers, is the axons. And the gray matter is the cell bodies of what?
Neurons.
Of neurons.
Right.
So here,
Wait, what was that? The white matter is...
usually the axons, and the gray matter is the cell bodies of neurons. So our next topic for discussion here is going to be the neuron. So in the quiz, let's go back there. I think now you're familiar with label the four lobes. I hope that all of you got them right. Okay. So number one here is for what? Frontal here. Parietal. Occipital. And temporal. What about the neuron? What are the different parts of a neuron? So this is the axon. All right. This is the dendrite, and this is in the middle is the cell body. Well, why is this the axon and this is the dendrite? Okay, good point. What else?
You travel down the dendrite through the cell body through the axon,
Mm-hmm.
and the flow of information stored in the axon.
Uh-huh. What else would tell you from this figure that this is the axon, not the dendrite? Yeah, exactly as Yasser said. But the other thing is that, what do you see here? There is a synapse. So the synapse, we'll speak about that, okay? So this is I would say a general view of what a neuron looks like in the central nervous system. You never, ever see a neuron like this, by the way. This is the perfect example of a neuron. But usually, generally speaking, neurons have three major components. The first one is called the dendrite, which is this tree of It looks like a tree that extends and merges towards what we call the soma, the cell body of the neuron, which then produces through what we call the axon hillock, what we call the axon, which is the pathway that connects the soma of the neuron to different neurons. Okay? However, there are like a million different morphologies of the neuron. Some of them have very extensive dendrites and have very teeny tiny axon. Some of them don't have dendrites that are extensive, very short, very limited, but have generally axons. Okay, axons can be single, can be multiple, cannot be there. So all these variabilities can be considered whenever you're looking at the morphology of a neuron in the central nervous system. And some examples on that, for example, the chandelier cell in the cortex or in the cerebellum, it's called chandelier because it looks like a chandelier. Okay? But how would you imagine a chandelier? It's like protrusions and then a long line that connects it to the ceiling, right? So this is exactly how this cell looks like, where you have the dendrites surrounding the cell body, and there is the long axon. Okay, there is the pyramidal cells in the cerebral cortex. They look like pyramids. There is the Purkinje cell in the cerebellum. It has the most extensive dendrite network of dendrites known in the central nervous system. Well, what we agree upon so far is that neurons process information. They collect, they process, and they give output. We spoke about the three basic parts, and they have a wide variety of shapes. Is the neurons the only type of cells that is present in the central nervous system? No. What are other types of cells that are present in the central nervous system?
Astrocytes.
Glia cells in general. What else? Okay, let's go a little bit beyond the book. Okay, we're saying, I already mentioned oligodendrocytes, or not mentioned, I already referred to oligodendrocytes as part of the glial cells. But what else? I mean, think about it. Red blood cells, blood. Why blood cells, right? Don't we have blood vessels as well? Don't we have linings of what we call the ventricles? Well, these are considered glial cells. Okay. So, neurons are not the only inhabitants of the central nervous system. There are many other types of cells. The most important of which to the central nervous system are called the glial cells. And by the way, the glial cells occupy more space than neurons do in the central nervous system. So it's majorly glial cells rather than only neurons. What are the types of glial cells?
Wait, are the glia on the white matter?
They support the white matter. You can tell that they support the white matter.
But white matter is the axon.
And white matter is fibers, exactly.
And what's the gray matter again?
The gray matter is cell bodies. Cell bodies of neurons, usually. Okay? So, who wants to remind me what are the different types of glial cells that you read about?
Oligodendrocytes.
Oligodendrocyte. What does oligodendrocyte do?
Yeah, the ones that are myelinated, that
They myelinate axons. Where? In the central nervous system. Well, are there myelinated fibers in the peripheral nervous system? Okay. And Schwann cells do that. Okay? So this is a particular useful piece of information. There are two types of the same kind of function that differentiates across the central nervous system versus the peripheral nervous system. So in the peripheral, we call them Schwann cells. They myelinate axons and lay down myelin. In the central nervous system, we call them oligodendrocytes. Okay? What other types of glial cells can you think about? Ideas? Astrocytes. Where do you usually find astrocytes in the central nervous system?
Brain?
It is in the brain. Where?
blood vessels.
Is it exactly in the blood vessels, or?
No, like,
Okay, it's usually astrocytes are usually surrounding blood vessels at a very close proximity to blood vessels, and they usually serve as an intermediate between neurons and the blood. They usually take nutrients out of the blood and transport it to neurons. They have very complex functions. Very little of that is known, and I can't say much about that. However, you can think about glial cells in general as supportive cells of neurons. This does not mean that they don't have their own functions that might support cognition, because there have been some studies showing that concentration of glial cells or types of glial cells can support function. However, very limited research, or not, I wouldn't say very limited research, but very limited evidence is available on that particular study. Okay. So we learned so far about different parts of the brain, generally speaking. What is the building block, or what are the building blocks of the brain, or the central nervous system, the peripheral nervous system? Well, how did people learn about that? So the neuroscientists used a set of tools that are used for neuroanatomy research. And using these tools, they realized, or they understood, the basic structure of the brain, the major types of cells, and later we would understand how they use these techniques in order to understand what is the function of particular parts of the brain. Okay, so if you want to know what are the constituents of the brain, what would you do?
Scan.
Take out a brain and look at it, right?
oh,
Is that always possible?
yeah, no.
I would say for humans, no. However, for animals, you definitely can do that with regulations. But for some very special people who donated their bodies for scientific research and for anatomy, dissection, and studies, these enabled us to understand further what the brain is composed of and what are the different regions that we're talking about here. Now, these are what we call post-mortem studies. So studies after death, and allowed scientists and neuroanatomists especially to do dissection and understand the different parts of the brain. However, recently there were some non-invasive procedures that were deployed in order to understand or to study the brain structure in living humans. One example is computerized tomography, CT scan, where using multiple X-ray images, one can build a 3D model or a 3D image of brain structure that enables us to study in detail the brain structure and the anatomy of the brain. In addition, there was another breakthrough of what's called an MRI, late 80s, magnetic resonance imaging, which takes advantage of the magnetic nature of blood with and without oxygen, and ultimately captures images from the brain that allows us to look deep, even better than CAT scans, than CT scans, and study further the anatomy of the brain. Also DTI, diffusion tensor imaging. This is a particularly useful method of imaging or looking at the brain whenever one wants to study the different tracts, the different fiber tracts in a particular brain structure. Okay, these particular techniques were very useful to study lesions in the brain that are related to particular brain disorders and understand further what are the bases for these diseases. So for MRI, as I said, it is all based on the magnetic nature of water and blood, and as a consequence, You all know what is an MRI machine look like, what an MRI looks like, right? You can go across the hole and you can definitely know more about it. So you get inside that tube, you are inside a big magnet. And whenever that magnet is on, all atoms in a particular area of the brain are aligned with that particular magnetic field. Then a radio wave is used to distort this alignment of vectors. We, as scientists, what we do is that we capture the time that it needs all these vectors to get back to their resting state, and accordingly, we can capture differential images according to the magnetic field that is produced there, and understand the different densities of different structures of the brain, which constitutes a picture or an image similar to that. And this is an MRI machine. It looks like this. So we get detailed 3D models of the brain. It is very safe and relatively low risk compared to other techniques such as CT scans, for example. You are exposed to a really large amount of radiation, but MRIs, almost zero. And this particular technique revolutionized the neuroscience research over the past, I would say, 20 years. Diffusion tensor imaging. It uses MRI to measure white matter tracts of nerves. It is particularly important in studying nerve tracts or fiber tracts within the brain. Particularly useful for disorders that involve deficit in fiber tracts in the brain. For example, one disorder is called MS, multiple sclerosis. What happens in this particular disorder is that the body attacks the myelin sheet that insulates neurons in the central nervous system. So what happens is that using DTI you can detect exactly where the deficits are and know how to respond. Okay, in summary, the major parts of the central of the nervous system are the central and the peripheral nervous system. The brain has 4 major lobes: frontal, parietal, occipital, and temporal. And neurons and glial cells are the building blocks of the central nervous system. Neurons are majorly composed of dendrites, somas, axons. There are different types of glial cells that support neurons and neuron function. And over the past number of centuries, there were multiple techniques and multiple procedures that were used to study human brain and understand more about its function and its nature, and ultimately served well to study further the function of the human brain. Okay. Up until now, we didn't say a lot about function. We just discussed briefly what is the motor function of the brain as per receiving signals, processing signals, and giving directions to the effector organs such as muscles and glands, for example. All right, let's apply what we already learned so far. Okay. Can you label the parts? What is the part in blue? Cortex. What is the part in light blue?
Corpus callosum.
Okay, the upper part is corpus callosum. What is this part? It is called the ventricle. What is the lateral wall of the ventricle? Okay, no problem. What is here? What is this? This is thalamus. What is this part? We said that it's like the main pathway of the brain, remember? The brainstem, the red, the cerebellum, the gray, spinal cord, okay. And what about the pink part? It is the pons. Excellent. Okay. Congratulations. You know very well about the brain right now. So what does the brain do? Okay, imagine that we have a cup of coffee in front of us, okay? Every one of you has a cup of coffee in front of you. What would happen in terms of brain function whenever you look at that cup of coffee and you want to grab it? Let's imagine together. What would be the first thing that happens there? Put yourself in that particular position and you see the cup.
You can sense and scan.
Exactly, before sensing that. Okay, I saw the cup. What do I want to do then? So what happened here? Input, processing, and did I do anything right now so far? Nothing. So I'm up until the point where I want to decide what to do. So do I want really to grab the cup of coffee or just stay away from it, or what should I do? Usually the response is I want to pick that cup of coffee, okay? But before doing that, what should I do? I want to make sure that cup of coffee is not extremely hot, right? Also, I want to make sure that I am holding the cup of coffee appropriately. I'm not really holding it, I'm not having a very firm grip. I don't want to spill it on myself, right? I want to make sure that all my muscle functions are coordinated enough to grab that cup of coffee, get it up to my mouth, and drink coffee, right? How does that happen? So, starting with the first step, when I saw the cup of coffee, sensory input through my eyes traveled through my what do you call it? Optic nerve to the area what we call the visual cortex and the occipital lobe. And then from the visual cortex, there were correspondences or messages between the visual cortex and the frontal cortex, which is responsible for decision making. Then from the frontal cortex, what happened is I made a decision and I wanted to move forward. So what happens is that the frontal cortex contacted what we call the primary motor cortex, which is also in the frontal area. Primary motor cortex is the one responsible to execute these orders, these decisions, sent these orders through what we call the internal capsule, which is the fiber that goes down the brain through the brainstem towards the spinal cord, and then to the muscle that moved. Is that enough? So I moved up until now here. Is that enough? That's definitely not enough, because I need to have that signal pass through what we call the subcortical regions, such as the thalamus, the basal ganglia, the cerebellum, in order to process that signal further and make sure that my movement is properly coordinated, to grab the cup properly, move my muscles properly, and move it up to my mouth in a smooth manner. Because otherwise, if it's all about muscle movements or muscular responses, I'll grab it, I'll spill it on myself. That's it. But it's way much more complicated than that. And that's what differentiates two major pathways for motor systems in the brain. The one we call the pyramidal pathway, that is responsible for this crude, I would say, brain to muscle orders. And then the extrapyramidal, which is all the refinement that takes place in the basal ganglia, in the cerebellum, and ultimately leads to me picking up the cup smoothly and achieving the ultimate outcome, which is drinking coffee. Okay? And it doesn't really stop here. So whenever you pick up a cup of coffee later, it becomes much easier. Why? Because your brain now knows the way. Okay? And this is, again, the importance of these extrapyramidal pathways that store what we call habits. And this is going to be the major, I would say, gist of this course, showing you exactly how cortical and subcortical systems interact to store a memory.
Repetition.
Sometimes it is repetition, sometimes it is not. So what is the capital of France?
What?
What is the capital of France?
Paris.
It is Paris. So this is a fact. When is your birthday?
April.
Okay, this is another fact. Okay, do you remember your 20th birthday?
No.
So even if you did, you would remember a sequence of events.
Actually, I do.
Okay, great. Okay, so this is what we call episodic memory. So what I'm trying to say here is that it's not only about repetition, it's about connecting a time, for example, with a context, knowing a fact, learning a habit, storing a number, for example, for a very short period of time. These are totally different types of learning. And we'll discuss that in detail. Hopefully Dr. Gluck will speak about them during the course. Okay. So as you can tell here, this is what happened. We generate behavior, pick up the cup of coffee, and drink coffee. Okay. This is what we call voluntary movements. But what about whenever you touch the tea kettle or the teapot, and then you notice how hot it is, and then you withdraw your hand very fast? What do we call this? It's involuntary. In other words, it is called a reflex. Okay? Why does a reflex happen? How does it happen? How does it take place?
Before you can consciously realize that it's hot, your brain knows and pulls your hand away.
Excellent. So it's not about the brain. It's about something that is way before the brain. We don't reach the brain actually in a reflex. We just go through a circle, or as I would say, a circuit that passes only through the spinal cord and gets back to the effector muscles. For example, have any of you ever do research with babies? Do you have all probably younger sisters and brothers?
I'm a midwife.
Oh, okay, you see babies all the time then.
Well, I'm not practicing now, but that's what.
Yeah, so what I'm trying to say is that there are multiple reflexes in a baby that you can see. For example, there's something what we call a suckling reflex. Whenever you put your finger in a baby's mouth, they start sucking. So this is the suckling reflex is I would say, primitive reflex that disappears later. There is also the palm grasp reflex. So what is really interesting about these reflexes is that they disappear once babies advance a lot. There's what we call also the parachute reflex. Whenever you hold the baby in the air, they form as if they're in a parachute. Whenever you pull them to their backs, there is the Moro reflex. So they're trying to increase their surface area in order to decrease the impact. These reflexes are in babies. You can also notice them in animals. But with time, they disappear. Why is that? Because they don't need them anymore. Our voluntary senses are evolving and giving us better control of our bodies. So why need that? And look at how strong these reflexes are. This is a newborn holding a small stick with a palm grasp reflex. And she's not even falling down. Look at that. So this just tells you exactly how powerful and important these reflexes are. Okay, how do they happen as we spoke earlier or discussed earlier? It's all about subconscious level. Doesn't really get to the brain. It's a circuit that gets into the spinal cord and goes back to the muscle instantaneously. So there's no delay because this is an emergency and you want to get your hand off the hot pot. You don't want to get hurt, right? Okay. So this brings us again to discussion of sensory pathways. So let's go back to major brain structures. So I call this area of the brain the front area, the frontal lobe, right? And the area that is next to it is called the parietal lobe. So generally speaking, are these areas distinct? Are they separate in the brain? Whenever you have a brain in front of you, are they separate? Well, they are separate. Okay, they have boundaries. For example, the frontal lobe is separated from the parietal lobe with what we call the central sulcus. So there is the central sulcus. The hemispheres are separated by the median fissure. Okay, so what I'm trying to say here is that there are distinct boundaries. So I mentioned the central sulcus between the parietal and the frontal areas. There is the Sylvian fissure here between the temporal and frontal and parietal. There is a parieto-occipital fissure here between the occipital and the parietal. So there are boundaries that separate these different lobes from each other. These are not only important landmarks that we can look at in the anatomy, but also important in functional regionalization of the brain. What I mean by that is, if you look here at the red area, this is what we call the primary motor cortex, or what we call precentral gyrus. So any of these foldings of the brain we call gyrus. So this is called the precentral gyrus, and it has been shown using recordings that the precentral gyrus is totally about motor function. So this is the primary motor area, while the postcentral gyrus is called the primary somatosensory cortex. So how do we know, how can we tell them apart? Using the central sulcus. Okay? So that's why it's really important to know brain anatomy if you want to understand behavior, because you want to associate whatever behavior you are observing with the structures, with the biology, with the transmitters that are responsible for the behavior. Okay, so apart from that, whenever we want to speak about sensory pathways, they extend from, as you can imagine, primary sensory cortex, go down, or go up. Did I use the correct sequence here? Is the primary sensory cortex an end or a beginning of the circuit?
Isn't it just a part of the middle?
It senses, so it should
Begin.
it should receive something.
Yeah. And then
So it's probably the end.
reaction. Oh, it's the
It's
end.
probably the end. So what I'm trying to say here is, just focus with me, because it's the sensory cortex that receives inputs from throughout the body and composites these inputs and forms the messages that are going to be shared with the rest of the parts of the cortex and ultimately make a decision. So it is the end part of the sensory pathway. What is the first part? A needle prick, a fine touch, a vibration, a change in the position of your joint. All these things are sensory inputs. Vision, but it's not somatosensory cortex, it's a visual cortex. Listening, it's auditory cortex. These are the end products. But what comes first is the sensory input that goes, for example, for sensations, for somatosensory sensations. What we're speaking about here, for example, pain. It's a receptor in your skin that transmits a signal to the spinal cord, and from the spinal cord goes up to your brainstem, to your thalamus, and to the somatosensory cortex. Then processing takes place, a decision is made and returned via the motor cortex. So the motor cortex is the beginning of the signal for the motor pathway. And then goes down via internal capsule, pyramidal, extrapyramidal systems, brainstem, spinal cord, and then to the muscle or to the organ. Okay? However, it's definitely different for hearing and seeing, smelling. So the difference is that these senses don't go through the spinal cord, but go directly to their associated areas. For example, the primary visual cortex is all about neurons that conduct signals from the eyes, the retina, through the optic nerve to the primary visual cortex, which is called V1. Okay. So let's go back to the example. Yes?
I just have a question. So the motor and the sensory are two different things. Because the way I thought of it was, you, like, say you touch something,
Uh-huh.
or whatever, you sensed it, and then it was integrated in your thalamus, and then the motor was the response. So the motor was the end, and the sensory was where it started.
Okay, that's a very good question. Actually, the thalamus is not the end point there. The thalamus is only a relay station. So what happens is that you get your sensory input to reach the thalamus. The thalamus sends it to the sensory cortex. Then the sensory cortex, with the rest of the cortex, particular areas that are definitely needed in that particular processing, are activated. The ultimate decision then is conducted to the motor cortex that through the thalamus, or not the thalamus, through the midbrain, conducts an order for muscles or organs to act. Then you withdraw your hand. Okay? So it's not at the level of the thalamus, it's at the level of the cortex, because it's a voluntary decision. However, just make sure that we differentiate between a reflex and a voluntary action. A reflex is only a circuit that goes through the spinal cord, does not get to the brain. Okay? While a voluntary action is something that you look at, you think about, it takes longer, but you make a conscious decision. Okay? Yes?
You said the auditory and visual go straight to the brain, or straight to the specific cortex.
Exactly.
They never go through the spinal cord.
They don't go through the spinal cord.
Kind of stuff.
Exactly. They go directly to the thalamus, areas that we call lateral and medial geniculate bodies for visual and auditory processes. And these are special senses, so I would say like they're definitely independent of the spinal cord. Okay. So,
Excuse me, Mohammed,
Yes.
are you posted on Blackboard by any chance?
I haven't spoken to Mark yet about it, but probably we'll make them available.
Where it comes in.
Maybe that's where the password comes. I'm not sure if these if the publishers make these slides available, but maybe we can send them to you as PDF. Okay. Yeah, definitely. But the most important is reading the chapter. Okay, you can edit that also, so you can take it out. All right. Okay. So let's go back to the neuron. So we spoke generally about the brain, about the function of the brain, about these circuits, how they integrate, how they, work in concert to produce, you make use of sensations and produce behavior or action. So how do neurons interact? How do they speak to each other? Okay, so we knew that neurons have three major parts: dendrites, cell bodies, somas, and axons. All right. It seems that neurons can speak two major languages. They're bilingual. The first one is called chemical neurotransmission, using what we call neurotransmitters. And the second language is electrical neurotransmission. Not only action potential, we'll speak in detail about that. So how does neurotransmission happen? So we mentioned earlier what we called the synapse. So a synapse is an area. Let me see if I have a slide on that. I think I do. All right. So the synapse is an area where two neurons come in contact. So we have a presynaptic element. So a synapse is a very thin area where two neurons come in contact. Okay? It's around 20 nanometers across the synapse. So there are multiple elements to the synapse. The first one is a presynaptic element, which is the element that is on the axon of the presynaptic neuron. And there is the postsynaptic element, and there is a synapse. Okay, so the synapse is a space that separates the presynaptic element from the postsynaptic element. So what happens in neurons, in neuronal synapses? What happens there is that there is chemical neurotransmission. How does that happen? So what happens in neurons in general is that when excitatory inputs get to the dendrites, they excite this neuron electrically. So inducing what we call an action potential, which is a burst of electrical activity that spreads throughout the neuron. And this particular, what's really very, I would say, characteristic about this activity is that it's all or none. So you don't have a half action potential. It's all either full-blown action potential or nothing. So there's what we call the all or none principle in action potential. So once there is an action potential, it goes through the whole dendrites, collects all to the soma, to the cell body. And then from the soma, through the axon hillock, through the axon, goes the whole way down. So what happens here is it gets to the point where the signal gets to the point where it is at these boutons, at these terminals, the axon terminals. With the activation of the axon terminals, vesicles containing chemicals called neurotransmitters are released in the synapse. Once they're released, they have corresponding receptors on the postsynaptic element that causes the postsynaptic neuron to activate or inhibit, or sometimes it's not only about electrical activation, but it's also many other things. It depends on the type of the receptor, the neurotransmitter receptor on the postsynaptic element that we're speaking about here. And it depends on the type of the neurotransmitter that has been released from the presynaptic element. Okay? This is what we call chemical neurotransmission, and which is precipitated by electrical neurotransmission through action potentials. Does that mean that neurons do not really connect to each other directly? Are there direct connections between neurons other than synapses? What do you call them? Gap junctions, okay. And it was a very big argument in the history of neuroscience between Ramon Cajal and Camilo Golgi, both of them were Nobel laureates in the early 1900s. And Cajal, the one who discovered Golgi apparatus, exactly. So Cajal was a big proponent of chemical neurotransmission, and that neurons are separate entities connected through something other than a continuous electrical signal. And while Camilo Golgi was a big proponent of what we call gap junctions. It was shown, actually, interestingly enough, Ramon Cajal was the one who was shown to be correct early, but then Golgi was shown also to have contributed extremely well to the neuroscience literature in this regard as well, because both of their theories were correct. However, the extent to which chemical synapses are used in the brain is much higher than neurons that use electrical neurotransmission. As I said, there are multiple types of neurotransmitters, multiple types of receptors of a neurotransmitter, and each one of these types of neurotransmitters has a particular function that was studied in the brain. So this is a table from the textbook showing different types of neurotransmitters with different types of functions that are associated with these neurotransmitters. For example, the most, the earliest neurotransmitter that was studied extensively was acetylcholine, which is very important in motor neuron function and neuromuscular function. We have dopamine, which is important for reinforcement learning. Serotonin, important for mood and sleep. Norepinephrine, important for arousal. Glutamate, the major excitatory neurotransmitter in the brain. And GABA, the major inhibitory neurotransmitter in the brain. So as you can see here, it depends on the type of neurotransmitter that we're speaking about. It also depends on the type of receptor on the postsynaptic element. Generally speaking, there are two types of neurotransmitter receptors. The first type is called ionotropic receptors. The activation of these receptors leads to passage of ion currents to the postsynaptic neuron, ultimately leading to excitation or inhibition of the postsynaptic neuron. So it is all about electrical activation via chemical neurotransmission. The other type is called metabotropic. And the metabotropic type of receptors does not only, some of the metabotropic receptors are linked to ion channels and lead to ionic activation. However, the effect of metabotropic receptors extends way beyond the synaptic activation, leading to modification of DNA translation, recruitment of internal cell machinery, and many other things that extend beyond the initial synaptic event. Okay. So, given that we know all of this about the brain, how do we know what is the function of each brain region? It's still a puzzle, so how can we tell? One clue is presented to us by the science of neuropsychology, which is studying brain function upon completion of a particular behavior. Okay? So, for example, we determine a particular brain area. We expose that particular subject to a set of behavioral measures or set of behavioral tasks to complete. Then, after completion of the tasks, we retest the brain area using one of the imaging techniques that we're speaking about. This is in humans. For example, in animals, what we do is we study the brain earlier. We can take out the brain, look at it, slice it, study under the microscope, then expose the animal, another matched animal, to the behavior and study the brain later and look at slices of pre and post behavior brain slices. And this way we can understand what is the impact of a particular behavior on the structure of the brain. Does behavior or behavioral activation change the structure of the brain? What do you think? Mm-hmm. It does. Okay. You can think about it also in the negative direction. So I was speaking about it as somebody who's doing a behavior, and this behavior ultimately affecting their brain structure. But you can also consider people who had brain lesions, and these brain lesions affected particular functions. One big example is patient HM, if you heard about patient HM, who had, these are the initials of a name of a very famous patient in the field of neuroscience and neuropsychology. And this patient had epilepsy and was operated on to remove the epileptic focus in his hippocampus. And ultimately the surgeons had to remove the whole hippocampus. That the patient HM ultimately ended up having a selective deficit in the formation of memory. So whatever happened after that point, after the surgery, did not really consolidate. All memories after that point were not stored. So using this particular example, we understood that the hippocampus, or the area that was removed from patient HM, was important for memory storage. This is another example. He's a very famous pianist. His name is Clive Wearing, and if you go on YouTube, there is a documentary on BBC on this particular pianist. And what happened to him is that he had encephalitis, viral infection of the coverings of the brain, and he ended up having structural deficit in the medial temporal lobe area. From that point on, he was unable to store new memories at all. However, interestingly enough, he learned piano, to play the piano, before he got the disease. So what happened is that he was fully able to play the piano, play all these very complex notes, but he wouldn't be able to tell you what this note is or who was the composer, despite the fact that he was one of the experts in the world about the composition of one of the very famous composers. I don't recall the name exactly. So this tells you exactly how different parts of the brain are involved in different types of functions. And sometimes, as I said, we can study that in a positive direction, where we can impose a particular behavioral set of tasks on an animal to do, then study the brain right after. or we can study patients with particular deficits, or induce these deficits in animals and study the effects of these deficits on behavior. Okay. This tells us, actually, there's a point that I should really discuss here, which is about Lashley's work. And Lashley is who wants to tell me who's Lashley? Who read the chapter? Okay, he's a neuroscientist who was searching early on in the 19th century, like I would say mid-19th century, and mid-20th century. He was searching for what is called the engram. Any idea what the engram is?
Like the center of the brain where learning will be stored.
Exactly, where memory. What is memory in the brain? So what corresponds to memory in the brain? Or the center, as you mentioned. So throughout his work, he was looking and making lesions in brains of rats and brains of animals, and trying different tasks, behavioral tasks. And the smaller the area he went to deactivate, there was no effect in the cortex. It was like his research was focused on the cortex. So he tried to lesion very small areas in the cortex and look at the effect of this lesion on behavior. But unfortunately, he found nothing. But when he did an extensive lesion, what happened is that the behavior was impaired. But it's an extensive lesion, so you can't tell which part of the brain is dead in the cortex. So he reached the conclusion that memory, or an engram, is not really stored in one place, but there are like multiple elements of a memory. Each of them is stored in a particular brain of a particular area of the brain. Hence, when you deactivate or lesion a large area of the cortex, you are impairing behavior. If you are only lesioning a small part of the cortex, it's only that particular part of the memory that is lost, but generally speaking, you don't affect behavior. Okay? And this informed our understanding of what a memory is, and it's partly it's parts of Lashley's definition of memory, and also the work of Gall, who found the science which we call phrenology. And what is phrenology all about?
Different shapes and different indentations in the person's skull tell, I guess, their personality and the way that they would learn and where different things would be stored.
Because the different shapes of skull would correspond to different modalities of development of different parts of the brain. And so the more prominent, for example, your forehead, the larger your frontal lobe, for example, okay? This was a totally erroneous view of the brain. However, if you think about the view of memory currently, right now, it's somewhere between Lashley's definition and Gall's definition, where memory is associated with particular brain areas, but also distributed across other areas. And the memory is a collective function rather than being a single function, but is composed of different components that are associated with different brain areas. So in summary, we spoke about the somatosensory and motor pathways in the brain. We spoke about how do neurons communicate through chemical and electrical neurotransmission. And we had a bird's eye view on how do brains produce behavior. And we discussed briefly some of the techniques that scientists used to uncover the functions that are associated with particular brain areas. Okay. Let's apply what we know, and then we will get a break. So botulinum toxin, commonly known as Botox, prevents action potentials from being able to release neurotransmitters into the synaptic cleft of neuromuscular junctions. How would botulinum toxin affect communication between neurons?
I mean, there would be no
Okay, what neurotransmitter are we talking about here? Acetylcholine, because it is a major neurotransmitter in the neuromuscular junction. Where else? Is it only the neuromuscular junction? How does botulinum toxin affect behavior?
It blocks the
It blocks the release of acetylcholine. Okay? So, what do you get in terms of clinical symptoms? Have you ever heard of flaccid paralysis?
Yeah, I know what
So this is exactly what is botulinum toxin would cause if your muscles or neuromuscular junctions are exposed to it. But what else? There is a major brain area that is called the basal forebrain that is majorly a collection of what we call cholinergic cells. And it has been shown that this is one of the major areas that are affected by Alzheimer's disease. So if I apply botulinum toxin, would that affect your memory formation? Would you do that? Definitely not. So what I'm trying to say here is it does affect memory, but not to the extent where we can study it in humans. Because there are like very few cases you see in hospitals these days. And however, botulinum Botox is used for cosmetic reasons. So how do people manage to prevent the adverse effects?
In the way it's administered, I don't know, is there a certain, I don't know, I'm guessing.
Okay, I want an educated guess. So,
Something is prohibited from,
well, if you give that in very superficial, not systemic, route, and apply that in very small doses, it does actually lead to filling up the gaps and removal of wrinkles. But if you increase the dose a little bit, all the other side effects are going to answer. All right, let's get a 10-15 minute break and then go to psychopharmacology.
that's great.
Go back to psychopharmacology. So in terms of psychopharmacology, it is the study of the effects of drugs on behavior as it acts on the brain. There are multiple mechanisms of action for drugs. For example, some of the drugs can prevent the release of neurotransmitters from the presynaptic element. Some other drugs can block the reuptake of neurotransmitters from the synapse. Some other drugs can block postsynaptic receptors. So depending on the mechanism of action of the drug, we can design studies that look at the effects of administration of a particular medication or drug on behavior. For example, if you look at heroin. Heroin is a narcotic, right? So what happens is that heroin is very similar to a substance that is produced in the body that is called endorphin. And heroin acts, but much more stronger than endorphins on particular receptors in the brain, leading to the activation and later dependence on this particular substance. So this is how we understood what heroin does to the brain, by applying this substance, or people who use this substance. Another example is cocaine. Cocaine is known to be a blocker of the reuptake of the neurotransmitter dopamine. So it impairs the transporter, what we call the dopamine transporter. This leads to a very high increase in the concentration of dopamine in the synapse, ultimately enhancing learning from reward. So this gives people who are on cocaine this like rewarding, pleasurable experience, not for long. We use this as well to understand the basic mechanism through which this particular medication acts on the brain and ultimately modifies behavior. There are always pros and cons to any science or any technique that we use. So some of the pros here is that it interfaces with the chemical language of the brain, and using different chemicals would ultimately result in different expressions of behavior and would give us the opportunity to dissociate the different roles of different chemicals in the brain. The cons include that there is no selective brain region where these drugs can act. So you just put cocaine into the system, the whole system takes cocaine, you don't know where it's acting, you don't know specifically where the drug is really playing a role, right? This is one of the cons. And the other important thing is, with time, there is what we call habituation. So, or brain becomes, the brain's tissue that is affected by this particular drug becomes more resilient, can accommodate that particular chemical more, and ultimately reducing the effect of that particular drug. For example, cocaine abusers take larger and larger and larger doses because the brain tissue gets used to that chemical, and ultimately they need a larger dose to get to the same level of, let's say, pleasure or reward that would get them where they would expect. Okay, what about, so we spoke about, this is a way to interact with the chemical neurotransmission in the brain. What about the electrical neurotransmission in the brain? This is the science of what we call neurophysiology. So neurophysiology is all about recording electrical activity directly from neurons. And you can imagine how difficult this task is. Finding a neuron, putting an electrode in that neuron, and recording electrical activity from particular neurons in the brain. In this particular experiment that you see the figure for here, an electrode was implanted in the motor cortex, the primary motor cortex in monkeys. And the monkey was instructed to move a joystick, right, left, up and down, and then upon moving of that joystick, electrical activity in the neuron that was tagged with electrode was recorded. And you can see here, according to the direction of movement, up, down, right and left, it was evident then that upon moving the joystick to the down direction, the firing of the neuron was the highest. In other words, it seems that this neuron is very sensitive to movements going downwards. Building on this, we can also assume that there are neurons that are sensitive to moving to the right, neurons that are sensitive to moving to the left. And it's an integrative function of all of these neurons that gives us the ultimate complex movements that we do every day. Whenever, let's go back to the coffee, the cup of coffee example. Whenever you're moving your hand to get the cup of coffee, you're doing rights and lefts and ups and downs a dozen of times here. So there are neurons, single neurons, that are responsible for every piece of this complex action that are activated upon the need for their action. Okay, so neurophysiology recordings were utilized to better understand the motor cortex in the human beings. So what happened is that there were patients suffering from epilepsy early on in the 20th century. And what happens is that surgeons offered these patients or asked these patients to record from their cortices while they're doing the surgery to remove the epileptic focus. Oh, well, you're asking me right now, okay, are these patients awake during the surgery? Why are they awake? Can you keep a patient awake during a surgery? Yes, we can if it is local anesthesia, but in general, can you? But why can we do it here? There's no pain receptors in the brain. So it's only about incising the scalp. After you're done with incising the scalp and getting through the bone, there's no pain. And you want to keep the patient awake because you want to make sure that all functions are intact. Okay? So what happens that some people or some surgeons use this opportunity to record from small patches of the motor cortex and understand where every patch in the motor cortex acts, or where every neuron of these neurons in the motor cortex is affecting. Ultimately, after huge effort in this regard, they got to form what was called the homunculus. So it is a human, looks like a human, but you see the proportions are really not representative of a human, right? But it's more representative of the amount of representation of each of these organs in the motor cortex. So for example, if you look at the hands versus the feet, so you see that the hands are way much larger. So what's happening here is that the hands are represented much more than the feet in the motor cortex. In other words, the area that is dedicated for sensation in the hands, or for motor function in the hands, is much larger than that for the feet, because the hands you need to find sensations in the hand. You don't need to find sensations in the feet. The same applies to the lips. The same applies to the tongue, but not necessarily the arm, not necessarily the shoulder, not necessarily the trunk. So in other words, these proportionalities correspond with how fine the sensory input or the motor input to these particular areas of the brain is. Okay? Is that idea clear? All right. And this is, generally speaking, the idea behind the homunculus, which represents the representation of these different areas of the body, how they map on the motor cortex, and some of them on the sensory cortex. Okay. We spoke about imaging techniques, we spoke about postmortem studies, anatomy techniques, we spoke about lesion studies, we spoke about single neuron recordings. What else can we do to study the brain? We said that neurons speak to each other using electrical signals. What else in the body uses electrical signals? The heart. How do we record activity of the heart? EKG, electrocardiogram. Well, if we place electrodes on the scalp, can we get electrical activity of neurons? The answer is yes. What do we call this? We call it an electroencephalography. And the output of that is called an electroencephalogram. So what happens there is that the ultimate output of the neurons is corresponding to these waves that are conducted through bone and skin and get picked up with electrodes placed on your scalp. However, are they in any way specific to any part of the brain? Let me be more specific in my question. So if I say that I have like 3-4 layers of cell types that are corresponding to point number A on the scalp, okay, right here. So I have like 4 levels. And level 1 is the cells are inhibited. Level 2, the cells are activated. Level 3, they're inhibited. Level 4 is activated. What would be the ultimate outcome on an EEG if I record it? What do we take in consideration here? So we have 4 different layers. One of them is the closest to the scalp. And ultimately, I would think about a vector that would involve the outcome of the electrical activity of these 4 layers up until this point. However, the influence of the closest area is going to be larger. So in other words, I would get a sense of what is very close to the scalp, what activity is going on there. However, it's to some extent inconclusive because it involves noise from many other areas of the brain. Okay? But it's a very non-invasive, very convenient, very cheap way of studying the brain function. You don't need to put together all these magnets in an MRI or PET scans or DTIs or CAT scans. These cost millions of dollars. But this is a very simple apparatus that you can use to record activity in the brain upon performance of a particular behavior, which is very convenient, but not very specific. However, we spoke earlier about functional neuroimaging. We spoke about neuroimaging. However, recently meaning like 20 years ago, we were speaking generally about structural magnetic resonance imaging, where you can understand the structure of the brain. But recently, a new way of looking at MRI scans was developed called functional magnetic resonance imaging, where you take scans of the brain before doing anything, like we call it the resting state scan, the baseline scan, and then after doing a behavior, what you do is, or throughout, during the behavior, during the task, there are acquisitions of images of the brain. So what you do later in order to analyze the images is that you compare baseline with performance and subtract the images. The subtraction of the images would filter out any areas that did not change between baseline and activity, and would highlight the areas that were either activated or suppressed due to that particular behavior. This is what we call functional MRI, fMRI. And fMRI is a very unique technique for studying the brain while doing the behavior, using a baseline image and an image during the particular behavior. This is very convenient and like very informative type of research. However, it's very expensive to do. Compared to EEG, way more expensive. However, it gives us the opportunity to look at brain areas inside the brain, way deep inside, and what they're doing during behavior. Another type of MRI is called PET scans, positron emission tomography. It is, in principle, it is similar to functional to magnetic resonance imaging, but instead of using blood perfusion, it uses glucose perfusion. So the fMRI uses the consumption of oxygen to measure activity. So the more active the particular brain structure, the more oxygen is going to consume. The same applies here, but in using a different technique, which is the more active the particular brain structure, the more glucose it will consume. And ultimately, different images are acquired, and sometimes different patterns of activity are shown. So PET scans are usually and fMRI acquisition are usually slower than EEG. EEG, you just record that electrical activity right away, you get instantaneous results. But for MRIs and PET scans, fMRIs and PET scans, it takes a while to process the images. It takes a while for the images to be captured, and this is one setback of using these imaging techniques. However, what is really nice in doing these functional neuroimaging techniques is that you get deep inside the brain, differentiating multiple areas in the brain rather than getting a rough electrical signal from multiple levels of neurons. However, recently there were new techniques that were developed where you can capture an EEG and an fMRI at the same time from the same brain, which taps into electrical activity as well as structural differentiation activity during behavior. And this is a very nice technique to use to study behavior in humans and animals. Okay. one of the controversies in the field is that some companies uses fMRI to tell you if you're lying or not.
That's what I was going to ask, if
Yeah.
there was a correlation with those.
They say so. So, but you know, usually I would tell you that scientists are very skeptic people. So,
But is that what's used in police stations?
no, not necessarily. That's a different. That's skin conductance.
It's,
Also, so you put a probe on the skin and you measure skin conductance.
Oh.
So usually, whenever you're stressed, your skin conductance is much higher. Okay, our final topic for today is called neuroplasticity. So let's say that we know right now we have an idea about chemical neurotransmission in neurons, how information is passed from a neuron to another using neurotransmitter. What if, let's say that I have 3 different synapses between the same 2 neurons, okay? What if I activate one of these synapses more than the others? What happens there? Does this synapse, does this connection become stronger? Does it become more reliable? How does this map into our understanding of what a memory is? So researchers have found that whenever you have we have a connection between 2 neurons, especially Heb, we call it Hebbian learning, the more you activate that particular connection, the stronger it gets. What does stronger mean here? It means that it needs a lower, I would say, generally speaking, lower concentration of that particular neurotransmitter to activate the postsynaptic neuron. So in other words, that connection, that root, becomes more potentiated. And this connection lasts longer, which means that if you think about it in the context of memory, this is, in other words, storage of this connection between these neurons, which ultimately would serve the purpose of probably recalling a memory. But is it only in the positive direction? I mean, like, is it only increasing? Is it only about potentiating neurons, potentiating synapses, connections between different neurons? So there should be definitely a counterpart that regulates these connections in the other direction, which is removing this potentiation. So the first part, we call it long-term potentiation, or the early parts of it, we call it potentiation, then it becomes LTP, long-term potentiation. However, there is another process that takes place, it's called long-term depression. So if I have 3 strong connections between 2 neurons and then ignore one of them, this particular connection will definitely become weaker. And here we're depressing the connection, and in other words, forgetting that memory, if it is a memory. Okay? This is a very short overview of neuronal plasticity, how it was developed, and what it means at the cellular level. And however, it's way more complicated than this, but it's not really the context of our discussion today. So this is only a general overview. So neurons and their connections are not fixed. They can be strengthened or weakened according to the use of these connections between neurons. And there are 2 processes that are very important in this particular aspect, namely long-term potentiation, which potentiates synapses, potentiates connections, and long-term depression, which does exactly the opposite. And I would urge you all to think about this in the context of memory formation and forgetting. Okay, I think that's the end of today's lecture. Thank you all for your attention. And
Thank you very much.
okay.